So last day we have learned about RBCs. Today we are going to learn about white blood cells, which are also known as leukocytes. They are divided into 5 types based on the type of cytoplasm. So we are going to learn about that in the next page. So generally the number of white blood cells in our body should be about 4000 to 8000 per millimeter cube of blood. But if it is more, it might be a problem, and if it is less, it might be a problem. So WBCs are actually associated with protection of our body from various types of toxins or virus or bacteria, whatever. So how do they do this? You are going to learn. So there is a process called diapedesis, by which the white blood cell moves out of any blood vessel, especially capillaries. They move out in the shape of an amoeba. As you have seen, you have studied in the lower classes, an amoeba moves in the with the help of pseudopodia, isn't it? So white blood cells will also show such a type of movement. They will come out of the tiny pores of a blood capillary, and this process is called diapedesis. And when they move out, if they encounter any bacteria or any other pathogen, they will just engulf it like an amoeba engulfs its food. So in the same way, white blood cell will engulf the bacteria. This is known as phagocytosis. This process is known as phagocytosis. So the process of movement of the white blood cells from the blood capillaries outside is called diapedesis, and the process by which they engulf the foreign material, it is known as phagocytosis. Next, let us see about the different types of blood cells, white blood cells. As I told you, it is based on the structure of cytoplasm. So it is divided into two parts, granular and non-granular. In case of granular, the cytoplasm is filled with small granules, small like dotted structure. But in case of non-granular, there are no granules in the cytoplasm of the white blood cell. Another difference is that in case of granular, the nucleus is actually divided into lobes. The nucleus is not a spherical body. In this case, the nucleus is divided into small lobes. As you can see in the figure, in case of neutrophils there are 3 lobes. In case of eosinophils there are 2 lobes, right? So this is a characteristic of granular WBCs. But in case of non-granular, there are no such lobed nucleus. It is one whole spherical nuclei. So now let us see in detail the different types of granular WBCs and the different types of non-granular WBCs. So the most abundant granular WBC is neutrophils. You have to remember it. Questions might come according to this, that which one is the most abundant granular WBC. So it is neutrophil. It has 3 lobes in the nucleus. This is another important characteristic. 3 or 4 lobes, 3 preferably, and its main work is to engulf bacteria through phagocytosis, like I told you, and it is produced in the bone marrow. As you can see, almost all the white blood cells are produced extensively in the bone marrow. Even red blood cells are also produced in the bone marrow. Next we come to eosinophils. So eosinophils decrease in number when people have allergic reactions in their body. Whenever a person faces allergy, which white blood cells will decrease in number? Eosinophils. This is another question which might be asked. So it has 2 lobes in its nucleus, and it also engulfs bacteria. It also secretes antitoxins. Antitoxins are chemicals which are able to remove the toxins from our body. They completely neutralize it. And they are also associated with allergy, like I just told you. They are also produced in the bone marrow itself. Next we have basophils. This is the least abundant granular WBC. Its nucleus is large, but it is not lobed into equal parts. As you can see, one lobe is very high in size and the other lobe is very small in size. So what is their work? This is very important. They release chemicals, histamines, for inflammation, which dilate blood vessels. For now, just read it. When you'll go to the higher classes, you will learn what is histamine, what is its essentiality, everything. It is required in cases of inflammation, whenever some part gets swollen. In that case, basophils play a major role, and they are also produced in bone marrow. Next is non-granular. There are 2 such WBCs. One is lymphocytes and one is monocytes. Lymphocytes is the smallest of WBCs, another important characteristic, and they produce antibodies. Antibodies are another another class of chemicals which help in protection against various types of diseases. We will learn about antibodies, antitoxins slowly as we move on to the chapter. They are also produced in bone marrow. Along with that, they are produced in lymph glands. I told you in the beginning of the chapter, there are a special group of glands, or there is a special system which is called the lymphatic system. So lymphocytes are also produced in those lymph glands. Next is monocytes. As you can see in the figure, its nucleus is in the form of a kidney, and their main work is to ingest germs. They are able to ingest, that means engulf the germs whichever enters into our body. Almost the major role of each of these white blood cells is the same for engulfing of bacteria or any other foreign material. They are also produced in the bone marrow. So this table you have to study nicely because it is very, very important. Next, let's see. We have already studied about this just now. Let us just do a recapitulation. What are the functions of WBCs? The first and the foremost is phagocytosis, coming out of the blood capillaries, and as soon as they encounter any foreign substance, they will just engulf it. This process is called phagocytosis. Now, what is inflammation? Inflammation occurs due to the reaction of tissues to injury and to localized invasion of germs. The inflamed spot has several characteristics such as local heat, redness, swelling, pain. As sometimes it happens that you get hurt somewhere and that part gets swollen, right? So what happens, that part might be a little hot compared to the rest of your body because of the pain and the swelling, and there is redness sometimes, right? So in those cases, which two leukocytes are important? Monocytes and neutrophils. Monocytes and neutrophils are essential. What do? They migrate through the walls of the blood vessels by diapedesis and fight against disease causing germs. They also destroy the damaged cells by phagocytosis. So whenever there is any swelling in your body, you have to understand that it is because of white blood cells. Whenever inflammation is there, white blood cells play a major role in reducing the inflammation by killing the germs or by killing the damaged cells. Okay, and sometimes we see that there is a pus formation in places where we get some wound. So that pus is mainly composed of the dead white blood cells which are dead now after working, and it also contains the tissue cells which were destroyed by the bacteria. So the white blood cells see that there is a bacteria present, they kill the bacteria, they die eventually, and that is that white liquidy part of wounds which is called pus. Formation of antibodies. So we have come to the topic where we will discuss about antibodies. Let's see what are these. The WBC, especially the lymphocytes, produce antibodies which kill or neutralize the germs or the poisons from them. So either they will kill the germs or they will neutralize it, or they will neutralize the poison which is released by those germs. Poison in the sense, poisons in this case are chemical substances itself which can cause harm. So antibodies are chemicals, or you can say antibodies are types of proteins which are produced by WBC in order to keep our bodies protected from foreign organisms. Introducing weakened germs or germ substances during vaccination stimulates formation of particular antibodies. I have told this to you in the first video that whenever vaccine is given to us, either it is a dead germ, either it is a weakened germ. So generally, if it is a weakened germ, there is a lot of problems which happen in our body as soon as we take the vaccine, for example in case of COVID vaccine, but that is actually producing the antibodies, so that at a later stage if we face the same problem again, we do not fall ill. So this is the reason. You will learn more about this in the higher classes. So we will learn about antibodies and antitoxins now. What are the differences between these two commonly mistaken terms? So as I told you, antibodies are produced by the white blood cells, and they are helpful in killing the bacteria or neutralizing the poison released by those bacteria. And these are chemicals, basically proteins, which circulate in our body, in the blood. And what are antitoxins? Antibodies are specific to toxins for which they are produced. The antibodies act as antitoxins which neutralize the poisonous effect of the toxins. So antitoxins are basically substances which will neutralize the toxins. So you can consider antibodies to be antitoxins. Antibodies are produced by the white blood cells and they act as antitoxins, and they neutralize the toxins produced by the bacteria or any other foreign material. So the terms are very commonly related, but they are different. So where are WBCs produced and what is their lifespan? The WBCs are produced in the red bone marrow, like we just learned, some cases in lymph nodes, and sometimes also in the liver and in the spleen. Their average life is very small, only about 2 weeks, and they die after engulfing bacteria or foreign material, and they are regenerated once again. Leukemia is a cancer of the tissue forming WBCs whose number increases manifold at the cost of RBCs. So I told you in the first video that blood cancer is generally associated with the increase in the number of WBCs. So how can it be problematic? You might think that increasing of WBCs mean more protection from germs. No, that doesn't happen. When there is an abnormal increase in the number of WBCs, it means that the RBCs are dying. And if there are no RBCs in your body, you cannot live, isn't it? No oxygen. So WBCs abnormal increase is also not very good. Sometimes what happens in diseases such as dengue and HIV, the WBCs decrease to such a low amount that people do not have any immunity left, and eventually they might die. So it's very essential that the amount of WBC is maintained at a particular rate. So next we are going to learn about blood platelets. As the name suggests, they are in the structure of small plates, and they are also known as thrombocytes. Their main work is for clotting of blood. Whenever we get a small cut in our body, blood does not keep flowing continuously. It stops after a particular amount of time. So this is done by the blood platelets. And also you have studied about the protein fibrinogen in the first video. So that fibrinogen or fibrinogen is very essential for this procedure also. Let us see about them. Clotting of blood or blood coagulation. When a blood vessel is cut, blood escapes from it. But soon a clot is formed on the wound and the flow of blood is stopped. This is done in order to protect our body from excessive blood loss. If it were not so, the injured person would bleed to death, like in case of various types of blood diseases. There are diseases such as hemophilia, where people's bodies are not able to clot their blood. It keeps on flowing and flowing. Okay, so those people can die at a very young age because of overbleeding. The injured tissue cells and the platelets which disintegrate at the site of the wound release a substance, thrombokinase. This is a substance, this is a chemical, or you can say this is an enzyme which is called thrombokinase. This is also known as factor X or Stewart factor. Stewart factor because it was discovered by a scientist whose name was Stewart. So whenever there are injured tissue cells, or there is an injury somewhere, there is a wound somewhere, the platelets at that part, in that part, they start releasing a substance called thrombokinase or thromboplastin or X factor or Stewart factor. The thrombokinase acts as an enzyme, and with the help of calcium ions present in the plasma, it converts a substance prothrombin into thrombin. Okay, so what happens in our plasma, in that white liquidy part which comes out along with blood, that contains calcium ions. So those calcium ions are very, very essential for blood clotting. So there are a lot of things which are essential for blood clotting. First of all, platelets. Second one is calcium. The third one we will learn later. So those calcium ions, it will convert a substance called prothrombin, which is inactive. It will convert it into an inactive form which is called thrombin. Next, let's see. Thrombin in the presence of calcium ions react to the soluble fibrinogen of the plasma to convert it into insoluble fibrin. So there is a lot of conversions going on. So initially it was prothrombin. Prothrombin reacts with calcium to form active substance thrombin. Now the thrombin, again with the help of calcium, it will convert fibrinogen to insoluble fibrin, so that there is a clot formation. Fibrin is a solid substance that forms threads in order to just close the wound so that blood does not flow extensively. These microscopic threads of fibrin are sticky and form a network at the wound. So as you know, whenever we have an injury, the first thing we do is to hold the place strongly with our hands so that excessive blood flow doesn't occur, right? So this can happen very naturally too. You don't have to do anything extra because our body already has the mechanism for that. As soon as there is an injury, the platelets start releasing thrombokinase, and the calcium ions present in the blood convert prothrombin into thrombin. The thrombin again reacts with the calcium ions and convert fibrinogen to fibrin. The fibrin form a mesh around the wound so that excessive blood flow can stop immediately. Blood cells are trapped in the network of fibrin. The network then shrinks and squeezes out the rest of the plasma, which is in the form of a clear liquid, the serum. I told you in the previous class last day that serum is actually the plasma which has no fibrinogen. So the fibrinogen which was present in the plasma, it has been converted into fibrin, and that fibrin has formed a network around the wound along with the red blood cells which had come out. As a result, we have a protective covering on the wound now. So whatever plasma is left now, it has no fibrinogen, so it is called serum. After a wound dries up, you will find a sticky clear fluid around it, so that is the plasma, that is the serum specifically. The solid mass which is left behind is called a clot or thrombus. So the solid mass which is present after the wound has started drying, that is known as thrombus. Okay, or slowly when it dries up, it heals, it is called scab. So this is how blood coagulation occurs. I told you last day, if you take a small amount of blood and you keep it in a test tube, you will find it to coagulate after a certain amount of time. This is very natural. It is because of the substances present in our blood, nothing else. So over here, as you can see in figure 7.5, when the blood has been left out, the clot is forming. It is a wrong notion that clotting is dependent on the exposure of blood to air. It is not essential that as soon as blood is coming in contact with air, a clot will occur. Clot occurs whenever there is friction, even or whenever there is movement of blood over a rough surface. Because sometimes what happens, you will learn this in the later stages of this chapter itself. Sometimes what happens, people who are very excessively fat, who are obese, they have a lot of cholesterol deposits in their blood vessels, so the blood flow might suddenly form a clot because of the excessive cholesterol, because blood is moving over a rough surface in that case. So clots can form inside our body too, in our brain too, in any part of the body. It is not essential that blood has to come out into the open in order to clot. If the number of platelets abnormally fall to a low count, then also coagulation occurs very slowly. Obviously, this is a very obvious thing. If the platelets reduce in number, the coagulation of blood will take a lot of time. This is also a disorder. This is also not good. Next, let's see the last topic in this part, which is called the blood grouping system. As you know, we all have different blood groups. A, B, AB and O. Negative and positive are there. So accordingly, if one blood is given to another person carrying a different blood, that might cause death. That might definitely cause death. But O blood group can give blood to every person. Similarly, AB blood group can receive blood from every person. How does it happen? What happens basically? A and B. Basically, there are two blood groups, A and B. A contains a special type of proteins on their surface, and B contains a special type of proteins on their surface. For example, let us consider it this way, that this is A blood group, A blood cell, and this is B blood cell. So in A, there are these type of proteins, and in case of B, there are circular proteins on the surface. Just imagine, this is just a diagrammatic representation. So the proteins on the surface are different, their structure is different, so their properties are different. In case of AB, every person who has AB blood group, half of their blood has A type of proteins, half of their blood has B type of proteins. So even if you give them A blood group, or even if you give them B blood group, there is no problem. Now in case of O blood group, what happens? If this is the blood cell of O, there are no such proteins. It is completely empty. So if there are no proteins, there is no problem, because the proteins will react with one another and cause a problem if it is given to some other person. So in case of O, there are no such proteins. So O can be given to any person. O cannot receive from every person, but O can give its blood to every person. Okay, so this is the thing. This is called the ABO system. You will learn more about this in class 11, but for now, this much is more than enough. So as you can see, these proteins are actually the antigens, and if two different proteins combine, they will definitely produce a fatal reaction which can cause death. Next, we have another type of blood grouping system, which is called RH blood grouping system. Positive and negative, like we say B positive, B negative, right? A positive blood group, B negative blood group. So what is this? We are going to learn in the next class. So we have the summary of the ABO blood grouping system. As you can see the figure or table 7.2, A can give to A and AB, but not to B and O, because B contains a different type of proteins and O does not contain any such protein or antigen. B can give to B and to AB. AB can give to nobody, because AB has both A and B. So if it gives to A, A will react with the B part, and if it gives to B, B will react to the A part, and if it gives to O will react with both the A and B part. So AB can only receive, they cannot give. But on the other hand, O is called a universal donor. It can give to anybody because it has no antigens on its surface, so no reaction. So O is called universal donor, but AB is called universal recipient. I'll send you a video showing that how if we give blood of a different group to another group, how does it react, how does blood clots, and the person might die because of that.